From the JAMA Network, this is JAMA Dermatology Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Dermatology.
Hello and welcome to this author interview from the JAMA Network. This is Dr. Adamson, the web editor for JAMA Dermatology. Neurofibromatosis type 1, also known as NF1, is a multi-systemic rare genetic disorder affecting an estimated 1 in 3,500 people. Over 90% of people with NF1 develop benign skin tumors that are called cutaneous neurofibromas. These neurofibromas can be associated with psychological stress, negative body image, and poor quality of life. However, there's a need to better understand which features of cutaneous neurofibromas most impact the quality of life in persons with NF1. In a new study in JAMA Dermatology, authors developed a large, crowdsourced, photographic, clinical, and genetic repository called the NF1 Genetics Registry to help measure the impact of cutaneous neurofibromas on the quality of life of persons with NF1. Dr. Kavita Sarin is a board-certified dermatologist and associate professor in the Department of Dermatology at Stanford Medical School in Palo Alto, California. She's a senior and corresponding author of this new article and is here to discuss this work and its potential implications for other rare disorders. Welcome to the podcast, Dr. Sarin.
Thank you, Ade. It's great to be here.
All right. So before we jump into this interesting study, you mind telling us a bit about your role at Stanford? Do you head a clinic for NF1 patients? How much research do you do versus a clinic?
Yeah. So I'm the director of the Skin Cancer Genetics Clinic at the Stanford Cancer Institute. In this clinic, I see a wide variety of individuals with inherited genetic conditions. Most of them involve cancer risk, so inherited melanoma risk, inherited basal cell cancer risk, and a wide variety of other inherited conditions that increase cancer risk. But we also see patients with rare genetic conditions, such as neurofibromatosis type 1, tuberous sclerosis, and so on. So we are managing patients with both rare genetic conditions as well as cancer predisposition syndromes. In terms of clinical practice, our clinic is run by me and an oncologist, Dr. Pauline Finchain, and it meets weekly on Fridays for this clinic, and I'm about 20% clinical time.
So you see a lot of patients that have or are at risk of developing certain types of cancer and skin cancers, and NF1 being no exception. Patients develop a lot of cutaneous tumors, these neurofibromas. That can be pretty bothersome. I guess this was the genesis of the study, trying to understand the quality of life related to developing these neurofibromas. Was there a specific patient experience you had? I mean, haven't we already known that these neurofibromas cause distress? Why did we need to study this further?
Yeah, that's a great question. And I think anyone who's seen participants or patients with neurofibromatosis type 1 know how critical these cutaneous neurofibromas are and how much of an impact the cutaneous neurofibromas have on their quality of life, social anxiety, mental health. They're itchy and they're painful. And so we know that these cutaneous neurofibromas can have an impact. This was perhaps most demonstrated to the nation in 2022, when I don't know if you remember the viral TikTok video where a young, brave girl with neurofibromatosis who was just riding a subway was photographed by a stranger, and the video was placed on TikTok and it went viral, and everyone thought she had monkeypox, and people said very harmful and hurtful things and even threatening things on this viral TikTok video. And then the New York Times covered the article, and the girl actually decided to bravely respond and say, I don't have monkeypox. I have a genetic condition called neurofibromatosis type 1. And I think that sort of showed the nation just a glimpse of what the daily experience of people with neurofibromatosis type 1 is, and how these, what everyone calls benign tumors that can cover their skin, are not really benign in their effects. So they may not be malignant, but the effects of these cutaneous neurofibromas can be quite devastating to people who have them. So we know this. And the question is, well, why study this? If we already know that cutaneous neurofibromas can cause a negative impact on persons with neurofibromatosis type 1, why do we need to study this further? And the end goal is because we want to develop treatments. We want to develop ways to reduce cutaneous neurofibromas. And in order to do that, we need to know what features of cutaneous neurofibroma should we target? Is it the size? Is it the location? Should we intervene on people with just a couple neurofibromas or people with hundreds of neurofibromas? What is the best time to intervene? Are there subtypes of neurofibromas that cause more itching or more pain? Are there locations of neurofibromas that have higher impact and so we should target first in terms of the treatment of neurofibromas? And so that was the impetus to the study. If we're going to develop treatments for neurofibromas, how do we think about what we treat in the cutaneous neurofibromas? How do we think about when we treat the cutaneous neurofibromas and who we treat?
So those are a lot of really big questions. Some of them are included in this study, some beyond the study. But this sounds like it was the genesis of what I think is probably one of the more important aspects of your study is how you were able to get this many patients to contribute so much information so that you can study quality of life related to neurofibromas. As you state in the article, this is a rare condition, right? Less than 200,000 people in the United States are affected, that being the definition of a rare disease. So it's challenging for a single institution in order to run a study like this. So you had to turn to other sources. Do you mind explaining how you came about using a kind of open source or crowdsourced means of recruiting more patients?
Yeah, that's a great question. And I think really highlights the challenges with studying rare genetic conditions is that these 100,000 individuals with neurofibromatosis type 1 are scattered across institutions across the United States. So any single institution may have a challenge in enrolling enough participants to be able to make meaningful discoveries into understanding how their cutaneous neurofibromas affect their quality of life. Collaborating with institutions is one approach. So we could have collaborated with multiple investigators, which we did. We collaborated with Johns Hopkins University, who has a large NF1 cohort as well.
It's a classic kind of registry-based way of doing things.
Yes, and there's many advantages to that approach as well. One of the disadvantages to that approach is that the measurements can be variable between institutions. So for example, me assessing my patients and their cutaneous neurofibromas may be different than another investigator assessing the cutaneous neurofibromas in another patient. And so we took another approach, which was to do a decentralized approach using photographs and developing a really robust registry of clinical data, photographs, quality of life outcomes, and genetic data. And so that we could study in a very comprehensive and cohesive approach how cutaneous neurofibromas affect quality of life. And the challenge for doing this is how are we going to enroll participants to participate in this study? And one of the things that was a surprise to me was how deeply passionate the neurofibromatosis community is about their condition and how receptive they are to researchers who want to study neurofibromatosis and advance research and development of therapeutics in this area. And so what we found was that we started reaching out to patient advocacy groups. And one of the largest groups we reached out to first was the Children's Tumor Foundation, which has thousands of individuals with neurofibromatosis type 1. And we thought we might be able to actually work with this advocacy group alone and recruit people with neurofibromatosis type 1 for our study. But what we found is that even though there's a large registry of participants with NF1, not everyone is interested in participating in research. Some people are joining for the community aspect. Some people are joining for education. But, we were able to recruit over 100 individuals who were interested in participating in our study. Not nearly what we needed, but certainly a large number.
What was your goal in terms of recruitment numbers?
Our goal was to recruit 1,000 individuals with neurofibromatosis type 1. And so we reached out to many different patient advocacy groups, patient community groups of people with neurofibromatosis type 1. And we've just found that across pretty much every group we reached out to, they were very receptive in wanting to help advertise our study, recruit for our study, and help in any way they could. As an example, one group, Patients United, made a video for us. They created the recruitment video to help with recruitment. Another group had posted our videos on social media networks. Another group found another investigator, independent investigator, to talk about our research and sort of validate it in their own community so that more people would participate in our study.
Patients are the most important part of doing clinical-based research, and it's amazing to hear that they were so engaged in trying to help in this endeavor and showing what neurofibromatosis, that experience, means to them.
And it was powerful, and getting the feedback was helpful too. We started hearing things like, we'd like it in another language. Not everyone speaks English. Can you translate into Spanish, or can you translate into another language? And that led us to actually change our study design midway. So we ended up translating into Spanish and German to be able to reach a larger population. We extended to additional countries outside of the United States to be more comprehensive in the people who could participate in our study. And we really relied on these 30 patient groups to really help advertise and recruit for our study, and for all of the participants to actually spend an hour of their time filling out surveys, submitting photographs, then submitting saliva samples for genetic testing. And we appreciate that this is a burden for each participant in the study, but they did this for the benefit of promoting research in neurofibromatosis type 1.
Another part of recruitment that you've mentioned is photos. So you had patients submit photos. And now, photo submission by patients sometimes can be a challenge because of lighting, because of it not being very reproducible, how the photos are taken. And you came up with a novel way in which to help make that a bit better. You mind talking about that very briefly?
That's a great question. And I think we should start with the challenges of photographs, because I actually didn't anticipate it would be so challenging when we first started collecting photographs. What we found was exactly what you described. There was a huge variability in the quality of the photographs, from people who submitted a picture of a single cutaneous neurofibroma to people who submitted photographs that we could barely determine their cutaneous neurofibromas because they were so blurry. So we did a couple of things. The first is we actually built a REDCap tool, which is an avatar-based tool to help patients determine how they should take their photographs. And this is an avatar of a human body with segments for their face, their arms, their legs, and chest. And every time they would click, for example, on the face, they could then submit a photograph of their face, and then their face avatar would change colors. So they knew that photograph was done. And they could do that for the various body parts. And that allowed a more simple and more straightforward interface for participants to be able to submit photos. And we actually made that REDCap tool publicly available because we felt like it was so helpful for the collection of photographs by dermatologic or skin researchers or any researchers who want to collect photographs of the body, that we put this on REDCap so anyone can download this as a tool for their study. The second thing we did is we needed our clinical research team to view every single photograph submitted. So every single photograph was manually reviewed for quality, for comprehensiveness. And if it wasn't up to our standards, we actually reached out to the participant and asked them to resubmit photos. And this was challenging for participants to have to resubmit photos multiple times. As you can imagine, it's extremely challenging to take photographs of your back, especially if you don't have someone else to help you. And we did require 7 body parts, face, arms, chest, back, and legs. And so that was quite challenging for participants as well. And that is a reason why, even though we recruited over 1,000 individuals who were able to submit very comprehensive surveys that take 30 minutes to an hour to complete, we were not able to get photographs from all 1,000 individuals to the quality we would like. And so for this study, we analyzed 583 photographs.
And so let's move to those results. You were able to analyze just under 600 patients that were able to complete all the various aspects in order to be included in the study. And you also counted each one of the neurofibroma on patients from these photos. Do you mind describing that process as well?
Yeah, so that's something we're currently doing through another method. But for this study, we didn't count each individual neurofibroma. What we did was we scored them. So we scored them into buckets of 4 categories. People who had less than 10 neurofibromas, 11 to 100 neurofibromas, 101 to 500, and then greater than 500. And that was partly because we didn't think we could be accurate enough with mobile photographs to get exact numbers.
You need to develop an AI tool.
That's on our list of things to do, and it's in the works. So we manually scored them, but we didn't score them just for number. We scored them for size. We scored them for location. We scored them for color. We scored them for subtypes of neurofibromas. So we had many features that each of these photographs were scored on that we wouldn't have been able to obtain just by a static text-based database. The other thing we looked at was, do we even need the photographs? Can participants self-report their number of neurofibromas? And we looked extensively at how patient self-reports of cutaneous neurofibromas correlated with investigator annotation of the photographs. And unfortunately, while there was correlation, we didn't feel like it was sufficient to be able to rely on self-report data. So we did choose to limit our analysis to the 583 participants, where we had 7 body sites for every patient.
And then probably one of the most important parts of this study is that you also gave patients a quality of life survey, the Neurofibroma Skindex, which evaluates various aspects, including emotion, physical symptoms, and some other things as well.
Yeah, so every participant filled out a Cutaneous Neurofibromas Skindex, which had 3 domains: emotion, symptoms, and function. And we were able to look at that as well as a visual analog pain scale and an itch scale as well, and look at how these cutaneous neurofibromas affected their quality of life and their symptoms. And we were able to look at how multiple aspects of the cutaneous neurofibromas affect this, such as, does this number of cutaneous neurofibromas matter? And at what point does it start to matter and affect their quality of life? Does the location of the neurofibromas matter? Does the color? Does the subtype? And at a level of granularity that we were not able to assess in smaller institutional studies.
And what did you find here? What are some of the main takeaways in some of these domains? I know there are a lot of them, but what are some that you found that stuck out to you most?
The first is that cutaneous neurofibromas affect all 3 domains of the Skindex: the emotional domain, the function, and symptoms. And so it really has very broad-reaching effects. The second surprising finding was that we found a strong impact even in participants with less than 10 cutaneous neurofibromas. And that was surprising to us because we really thought that we would see this impact primarily in patients with extreme numbers of cutaneous neurofibromas. What we did find was that as the cutaneous neurofibromas increased in both number and size, there was a much higher impact on quality of life, but we found that there was an impact even in participants with less than 10. Maybe less surprisingly, cutaneous neurofibromas present on the face had the highest impact on the emotional domain of the quality of life score.
I think the findings from the study are really important, and especially amazing that you were able to recruit so many active participants from around the world. But this also begs the question, now that you have this large cohort of participants, what's next? What other kind of work do you want to do to try to better understand the lived experiences of these individuals?
That's a great question. And we really believe that this resource that's been developed is going to be really transformative, both for our research, but also we're hoping to provide this to the general NF community of researchers to be able to use this to study phenotypes or conditions that they're interested in as well, in neurofibromatosis type 1 beyond the skin. But for us, what we're most excited about is that in addition to the photographic and the clinical data that has now been well phenotyped, we also have genetic data. So every one of these participants in our study has whole genome sequencing data from saliva samples they submitted. And so one of our next goals is to identify genetic modifiers or genetic variants that contribute to the development of cutaneous neurofibromas in their number and size. And so we'll be conducting genetic analyses to understand how variants in specific genes or regulatory variants may impact the development of cutaneous neurofibromas.
I'm Dr. Adamson, and I've been speaking with Dr. Kavita Sarin about harnessing crowdsourced data from adults with NF1 to understand the impact of cutaneous neurofibromas on quality of life. You can find a link to this paper in this episode's description. To follow this and other JAMA Network podcasts, please visit us at jamanetworkaudio.com or search for JAMA Network wherever you get your podcasts. This episode has been produced by Daniel Gusisi at the JAMA Network. Thanks for listening. Catch you on the next episode of the JAMA Dermatology Podcast.